nucleus, and then EU means true. True kernel means you have a true nucleus. And these are the two major separations of living organisms on the planet that we have. So if you were an alien and you take samples, you'd go either you're a prokaryote or a eukaryote. And you start going through the world, putting them into one or two possible categories. We will expand the prokaryote category later on. Right now, it's just the bigger world. Then we can look for some similarities, things they have in common between prokaryotes and eukaryotes. Something called a cell membrane, both of them have. They both have cytoplasm, and we can break that word down to help you remember what it is. Cyto refers to cell. Plasm means the fluid inside. You guys have heard of blood plasma? Or blood plasm? That's the fluid inside your blood vessels. So cytoplasm is found in both. They have something called a ribosome. They're found in both. And they both have DNA. For the DNA, though, it's different. Their DNA is it's circular, but it's also not found in the nucleus because they don't have a nucleus. Eukaryotes do have a nucleus. Looks like this purple baby's bassinet in a sense. And the DNA is in the nucleus. That's the big difference. Yeah, they both have DNA, but yours is not in the nuclear envelope. There are some other little bits of differences with these guys. If we go to the next slide, all right, here's kind of a cartoon else that prokaryotes have that most, not all, but most eukaryotes do not have, is this structure here called flagella. They're basically like propellers and allow the bacteria or the prokaryote to move through its environment. They can be found anywhere on the body of the prokaryote, and you use that as a biologist to figure out what kind of bacteria it is. Some are totally surrounded by it, and others just have one on the end. We have a very similar looking like structure in our anatomy if you're a male. This is kind of the general pattern of a sperm cell. Right? They have the body and a single flagella, which allows the sperm to move through the environment. What these guys also have that all eukaryotes do not have is this thing right here called a capsule. It's another layer to their structure. Both prokaryotes and eukaryotes, all right, will have this thing called the cell membrane. We're going to look at that this morning. All prokaryotes will have this thing called the cell wall, and some eukaryotes have a cell wall. Like if you're a plant, you're going to have one of these too, and plants are prokaryotes. So in other words, everybody has a plasma membrane, which is the same thing as a cell membrane, just a different word for it. Everybody has one of these. All prokaryotes have a cell wall. Some eukaryotes have a cell wall. Only prokaryotes have a capsule, the third outer layer. The capsule is really helpful if you're a bacteria. It's what allows you to sneak through your host's immune system and not be detected in a lot of cases. So you've got the cell wall from the inside out. Let's go that way. Plasma membrane, cell wall, and the capsule. 3 layers of defense to keep your DNA protected from the environment. We'll get a little more into this a little bit later. But some prokaryotes, if the environment becomes unfriendly, will shrink on down to another layer or another type of structure. You may know what that structure is that allows them to survive for a long period of time. Spores. Right? Bacteria can become a spore-like structure. And that's basically their version of a hibernation state. So if things get really crappy in your environment, you'll survive by turning yourself into a spore, or they're known as an endospore. And then you can survive. And we didn't really appreciate how long that could happen until, bless you, until a few years ago with advancements in technology, we cracked open a meteorite and found bacterial spores that were hundreds of millions of years old. And then they came back to life on Earth. Another interesting idea about where our life came from, perhaps. Which means they can survive space for a long, long, long time. Which means they can survive our environment on Earth. So, you know, you can make a really good horror movie where you're an archaeologist and you go into an Egyptian tomb that's been sealed for 6,000 years and you awaken some deadly plague because you now cracked the seal to the building and you provided a good environment for the endospore. It goes, oh, there's food and life out here. Let's reawaken ourselves, and then we'll go through the world spreading a plague and we'll kill everybody. All right? That could happen. It's one of the scary things about anthrax. It can last for decades in a spore-like fashion. So if you kick up the dust, now you're spreading the anthrax into your lungs, you'll breathe it. Oh, it's moist and warm in here. We'll reawaken and flourish. We'll learn more about the little nice, nasty guys later.
You said that prokaryotes, like pro means before and the karyot means kernel.
Kernel, which refers to the nucleus.
Yeah, did you give the one for eukaryote?
Yeah, EU means true.
True.
So that means you have a true kernel. That means you have a true nucleus. All right. Now, if you were to look at this picture and I were to say, tell me what kind of a cell this is. Is it animal or plant? You'd go, I don't know if I'm taking your class, John.
Prokaryote.
All right. We can look at this and go, okay, well, I'm looking for things that tell me if it's animal or plant. So I can see the plasma membrane. That's cool. Got that. It has what's called a cytoskeleton. All right. Cyto, remember, means cell, like cytoplasm. And it's a skeleton, like you have a skeleton to keep your form, to keep your shape. So cells have a skeleton in a sense, right? That's made up of cables and to survive in harsh environments. And it is still dying in our office because we keep forgetting to water the poor little thing. I'm the only one that really waters it. I'm the animal person in that sense. Like, that's really ironic, okay. But this is why you want a vacuole if you're a plant cell. You can also use it to store water. So when you water your poor wilted plant and it begins to stand upright again, sort of like the scarecrow, it's because it's sucking up all that water and storing it, and this swells up, creating pressure, which allows it to stand up. It's kind of cool. Chloroplasts are involved in making energy for the plant to thrive off of. And you're going to learn how plants do that in several weeks from now. Actually, not even several weeks. In about a week. All right, so if you can find a vacuole, chloroplasts, cell wall, you're looking at a plant cell. Animal cells do not have those things.
So you said the chloroplast was the energy?
It's going to help the plant turn sunlight into sugars, make energy. All right, let's look at this thing called a plasma membrane or a cell wall to understand how it's structured. Remember we looked at macromolecules. I said don't memorize the actual structure of it, but we had this thing called a phospholipid that I wanted you to make note of. This is a phospholipid. It has a choline, a phosphate, and then there's your glycerol with 2 fatty acids. So, little pop quiz. Be brave, raise your hands when you know the answer. Is that molecule, is it hydrophobic or hydrophilic? Is it hydrophobic or hydrophilic?
Hydrophobic.
Why?
Because it has more than 6 carbons that can't break apart or dissolve into the body.
That's a really good rule, right? In other words, it's a big molecule, right? And most of it is non-polar. But that end is polar. So, in a way, there's a new word we can give you guys. Amphipathic. It has a hydrophobic end, and it has a hydrophilic end. It's both. It was a trick question. Oh, I didn't create the trick question. I just have to teach you about the trick question.
But it's confusing.
So remember what makes something hydrophilic? Yeah, it's polar, right? Well, look at all the charges that are up here. There's a plus there. There's a minus over here. That means that part likes to interact with water. But then there's those long carbon chains that are hydrophobic that don't like to react with water. That's what makes a cell membrane so doggone awesome. The outer part of the membrane is this part right here. That's these little round balls that you see, the spheres. That part likes to be in water, but the long tails don't like to be in water. And they're called the fatty acid part. Now, I do a lot of TV listening as I'm grading papers. I'm always looking for examples to show you guys. And there was a TV show called, oh crap, what was it now? I don't know how to find it. I think it was How the Universe Worked. Your bathtub, half full of water, and stir it around really quickly. All right, they're going to organize themselves in terms of like things like to be with each other. These, look at the tails. See how there's tails here? And if you look over here, these guys are upside down compared to these guys. So if this is my phospholipid, I have a whole beaker, but I stir them all up, they orient themselves so that these guys try and line up together. And they'll do it and form a sphere, a big hollow ball. That's the same structure as a cell. This is our running hypothesis as to how cell membranes were early formed. Because now if I got a cell ball, and there's stuff inside the ball, it's isolated from stuff outside the ball. In chemistry, that's a cool thing. You have a separate environment now to do chemical reactions compared to the outside world. And that's the neat behavior of phospholipids. They naturally have a tendency to form what's called a micelle, or a big spherical structure. That means then that this really big, thick part, right, where all the tails are, is hydrophobic, but the round spheres, the heads, right, are all hydrophilic. And in case you didn't realize it, when I asked you guys if the compound is hydrophobic or hydrophilic, the answer was staring at you on the screen.
That's what was so confusing.
It gets both. Okay?
Seeing both on the screen, I'm like,
When we put a bunch of
should I-
group, phosphate group,
That
like
makes
glycerol to fatty acids, so basically, automatically those things form together as phospholipid, like a sphere, like a space.
Yeah?
It's automatically formed a sphere.
Yes.
Just put everything.
Automatically.
Oh, I got it.
Like I will automatically always go to a table full of cheeseburgers. But also we talk about it in terms of its structure. This is known as a phospholipid bilayer. Phospholipid bilayer. All right? And break that word down. Bi means two. Two layers. There's this layer of your phospholipids up here, and then there's the bottom layer down there. All right? This is the part that I'm going to really make sure you guys get. This is the part that you really need to learn and know. This is why Bio 160 is the prerequisite for Bio 241. Here you're supposed to learn the structure of cell membranes, how cells function, and what can and can't get in and out, and how they do all of that. So when you get into 241, you already have a pretty good idea of how this works. All I got to do is remind you and shake the cobwebs if I have you in Winter Quarter.
So if it's not a cell membrane, it's just an oil in the water, will it still act like this?
So if there's not a cell membrane, then really you have a big pile of goo. And there's no overall uniform structure to it. So you have to have a cell membrane to be labeled as a cell. So without a cell membrane, you're not a cell.
So if the phospholipid also have in the oil,
Have a which?
oil.
Oil. Act as a binding site. Now there's a couple of ways to do this, and I wish the artist had done this. The binding site could be for things like neurotransmitters and hormones. And that's how cells talk to each other, right? They send a message in the form of a neurotransmitter or a hormone, and it tells that cell what it needs to do. But it can also be a binding site for recognition. In other words, for your immune system, where this thing is going to say, I belong to John. I belong here. And if I had this receptor that didn't have my name tag attached to it, and any of your white blood cells or immune cells came by and noticed that a binding site that's known as your name tag in a sense, without a name on it, it's going to suspect that it's actually an intruder. And it will then destroy that cell or cause the cell to die. We're also then going to have, right, some proteins. And this is like, well, it's not really accurate in one way. It is a receptor, but it's also a channel. It's a pathway for these little green spherical structures to actually go all the way through to get to the inside, or vice versa, to have been in the inside and go all the way through. So you can label this as a channel / receptor. Depends upon actually what kind it is. So we're going to call them channel / receptors. This is just a channel. It doesn't have any receptors. So in other words, like a tunnel, it's always there. That will allow stuff to move through the cell membrane in either direction. It can go from the inside to the out, or outside to in. That's why they kind of call them transport. I don't really like the word communication too much, but it can be working for that. What I want you to really focus on is the idea of receptor / channel for these, and these just being channels. Don't worry about the labels up here for B, C, and D. This one I have no problem with, but the others like, well, you're kind of misrepresenting what those things are.
So I get the channel, I get the channel part, but what's the receptor part?
Well, so FYI, don't worry about this right now, all right? If you're one of these things, now what you call a receptor / channel is that some of your channels are always open. We're going to call them leaky channels. Some are like that door. They could be open or they could be closed. Well, if they're closed, I want them open. So they need to receive a chemical signal to cause them to open. So the receiving of the chemical signal is the receptor part. It's like when you go through a toll gate, right? You pay the guy the toll, he allows the gate to open up. Well, he receives the money, then he opens the door. You need to receive a signal to open the door.
So for B, you said you don't want us to go by recognition, but instead by binding site?
By binding sites, yes. All right, now